are great. He's a great scientist, but you'll be surprised. He's also a stunning photographer and a vivid bird watcher. So you can talk. I am impressed to have such wonderful hobbies. The great thing is that Greg's scientific career really started here at KU when he joined young and a junior faculty at that time, Bob Dunn, and Greg did quite a lot of research, and Bob's, you guys published a lot. I think your first scientific publication was here in 1997 with Bob Dunn, but ultimately. Greg graduated from electrical engineering in 1999, right? Correct. So his passion for spectroscopy that he probably learned from Bob from spectroscopy and knew at that time microfluidics, and I found the outlet where you joined a group of Professor Albert Falk, right, at the University of Washington. And you guys published very impactful work on microfluidics, on the developing field of microfluidics in PDMS, I might say, but nevertheless.
It still works.
There you go. In 2005, Greg earned his PhD in bioengineering from the University of Washington, and then joined NIST, and has been working at NIST since 2007. I'm impressed. I mean, Greg collaborated with numerous fantastic researchers. For you in the microfluidic field, you should hear about the bioengineer and analytical chemist Lori Lacascio, for example. We published with and planned working on single cell analysis using light microscopy. Then in 2016, Greg became a leader in NIST. This research focuses on creating microfluidic tools to improve measurements and enable discoveries in the biomedical field. His group is developing new approaches to measure optical properties of cells and material flow. Greg authored 70 plus publications. In 2024, was recognized for his work was recognized by the Department of Commerce Gold Medal and the Ron Brown Excellence in Innovation Award for the development of the serial flow cytometry. So today he will share some of the results on that very topic in the title New Frontiers in Flow Metrology and Cytometry from Combination of Microfluidics and Uncertainty Quantification. Thank you.
Thank you, Maggie. Well, it's awesome to be back here at KU. I don't get back enough, but I do get back regularly. So thank you, Steve and Maggie, for the invitation to come. Thanks, everyone, for joining me. As Maggie said, I started my research journey here at KU in 95, I think just after Bob had started his program in near-field scanning optical microscopy. Let me get my share going here. Okay. So real quick, because I work for the federal government and people care a lot about the measurements we do, I'm not here to endorse anything or to label any specific products as the best or most practical for their use. I'm just going to tell you about what we've done and the measurements we've made to analyze different performance and develop new capabilities in measurement science. So I started here at KU back in 95, working with near-field scanning optical microscopy. Got our first paper with Bob developing bent near-field tips. So my first fabrication experience, Bob had a good laugh about this because I didn't know how to make anything except out of wood. And so I built a 2x4 based hinged device to melt this optical fiber over a tantalum wire. left here then, graduated in 99, started my research in micropatterning cells at the University of Washington, actually with Viola Vogel. Did my thesis with her. She's now at the ATH and Zurich. And then for my postdoc, I was just with Albert for 1 year, so I got a real whirlwind in microfluidics during that time, and that's really where things took off. So I'm at NIST now, and I've been at NIST since 2007. As you may or may not know, NIST is the United States National Metrology Institute. So we are tasked with maintaining all of the definitions and comparability of measurements within the United States and standing those scales up internationally. So you can imagine we're in the Department of Commerce. Anytime a quantity is bought or sold or traded, people care very much about what it is, how much it costs, right, and how much the quantity is that you're trading. So that's why NIST primarily falls under the Department of Commerce. And we're also tasked with developing standards, anticipating new technology, and really thinking about the fundamental measurement questions that are hindering the advancement and production of U.S. Science and manufacturing. So George Washington anticipated this all the way back in the 1700s when the country was being founded, but it wasn't until 1900 that NIST was established as the center of weights and measures in the United States. And I think, a nice quote from one of our former directors from my early time at NIST is, if you know how to measure something, you can improve it. You can understand it, you can improve it, and you can move the field forward. So that's really the way we like to think about things. And, related to the fundamental measurements, we have time and mass, temperature, length, and illumination, illuminance, all measurement quantities that are defined and in many cases led within the world accuracy by NIST. So we take our measurement science and those 7th and 8th, 9th digits of precisions quite seriously. In some cases, we're nowhere close to that in biology yet, which is why I think biology is such a fun field to be working in, not just because it's so cross-disciplinary and unique applications of technology in new ways, but because biology, I think, really could benefit from better measurements. And so that's part of the reproducibility crisis, I think, is understanding how do we make better measurements, what do we know about the values we're getting, and how do we interact in such a way that we can repeat and reproduce each other's experiments. So within health and bioscience at NIST, my division and my group in particular are applying a lot of these new measurement advances in photonics and microelectronics and mass and fluid transports, thinking about bio and nanomaterial metrology, and then developing eventually standards and reference materials in support of those. And of course, those fall into application spaces and instruments and medical diagnostic capabilities for different technologies that many of you are familiar with. And I'm a microfluidicist now. And so I like to think about the advantages of using microfluidics to do these experiments. And, they're unique conduits for mass transport, so we can control the location and partitioning and dynamics of interactions of chemicals and materials on a really precise scale. There's some useful physical constraints. So we have boundary conditions and diffusion and inertial forces and other things that kind of surprising that happen on the nanoscale and microscale that you can take unique advantages of. Of course, we have very small volumes and high surface to volume ratios. So again, in chemistry, that's often an interesting property that we can utilize for experiments. And then we can do dynamic processes. So because the volumes are so small, it can be quick to interchange materials and reactions to study dynamics. So 2 things I'm going to talk to you about today that ongoing work in my lab. We developed what we call the nanoflowmeter, which is the world's most precise and accurate measurement of flows using optical properties, and then the flow cytometer for particle measurements that we've developed using microfluidics and integrating optical measurements. So let's start with flow metrology. So why do we care about flow so slow that we're talking about draining a can of Coke through your hair if it was a straw over 1,000 years, right? This is really small. We're talking about the volumes of cells over seconds, right? Why do we care about these applications? And some of it's still anticipating new technology, particularly in support of calibration of medical devices. We're perfusing drugs at the nanoliter per minute scale, but we're really not going below that yet, but potentially with more potent drugs and more specific refined precision delivery of these drugs, we might think about measurements below, several nanoliters per minute. Certainly thinking about safety and efficacy of microfluidic devices. When do these experiments leak? When are they failing? In a lot of cases, it depends on really precise measurement of flow to know that the device has stopped working. And thinking about improvement in the resolution of different technologies. So HPLC, for example, the resolution is often set by how fast you can drive the flow. Somehow I've got automatic advance here. I'll try to hope that doesn't continue. And then thinking about, reducing reagent consumption and improving sensitivity or specificity of how you're delivering fluids to reactions. Also, consistent, continuous manufacturing. So if we're continuously producing something such as microdroplets that we might polymerize, or if we're putting reagents into these droplets to do controlled high throughput experiments, we want to be able to understand what are the variabilities in the volumes and concentrations that we're partitioning in these droplets. So I think there's real application space there. And we're also looking at these to validate new physical phenomena and to understanding limitations in device technology where we're really up against that boundary between convection, so active motion of particles by mass transport, and then just random diffusion. So conventional technologies, I'm going to move through this pretty quickly for measuring flow. All of the National Metrology Institutes maintain the calibration of flow based on a very sensitive bucket and stopwatch. So you've got a microbalance and you've got a timer, and you are measuring the accumulation of mass onto that very precisely by thinking very carefully about adjusted buoyancies and stick-slip interactions with micropipettes interfacing with those weigh pans, as well as evaporation and other physical forces that start to dominate on that scale. The most common commercial device is based on heat transfer. So you have a heater inside a microcapillary and you have a sensor downstream, and you're just calibrating that transduction of heat within that over time. But of course, those, you've got heat losses that don't have anything to do with the flow. And so that really limits your ability to dynamically and carefully measure small mass flow rates. Front tracking is kind of the state of the art in the way that other national metrology institutes are thinking about measuring flow. So you have an interface that you control with very careful surface chemistry, and you measure the transfer of that interface over time under a microscope, and you can get very accurate measurements of flow. But I propose to you that that's not particularly dynamic. It doesn't last very long, because as soon as that interface moves out of your field of view, you no longer can measure mass flow. And you can't continuously measure anything interesting, because every time you reintroduce that interface, you're changing the material properties of the fluid you're flowing. So not particularly useful, I think, for real applications of flow metrology. And then, of course, you have particle image velocimetry, where you're measuring particles moving and mapping out the flow trajectory in a pipe in real time. But that's particularly computationally intensive. And as I sort of alluded to, as we move these technologies to smaller and smaller flows, the uncertainties associated with those measurements get exponentially large. So we're pretty good at measuring flows down to about 100 nanoliters per minute with flow metrology, gravimetric methods, bucket and stopwatch. But below that, our uncertainties get very large very fast. And the other thing to think about here, too, is that the dynamics are really slow. At 100 nanoliters per minute, it takes 5 minutes to make one measurement. So you're not measuring anything dynamic. You're only estimating the average flow rate over that time. So our idea was to measure flow using an optical system. And this is where we come in with our microfluidics. So we have a flow channel here coming left to right across an optical interrogation region. We have laser light that we deliver through optical fibers through an internal waveguide here to deliver light to a specific region of interest on the microfluidic channel. We can measure the transmitted light through that interrogation region, so we can normalize to the incident power of light. And we can also measure obscuration. If anything moves through, it will block light. We think about that absorbance. But So how does this work? Which is a 1 meter tall column of water, and we spike that water with a fluorescent indicator. In this case, we use fluorescein, primarily because it's a bad fluorescent molecule. So we're going to use the properties of fluorescein as the indicator of the flow itself. We control that stage to control the flow linearly based on the height of the water column. We've got very good control to within 2 micron, absolute precision of that, so 2 parts per million control and flow. And we are controlling the conductance of the system by the fluidic resistance of the chip. So we define a chip that has a pretty high fluidic resistance, about half a meter length of a 20 micron wide channel, to ultimately get to a conductance magic number here. We get about 1 nanoliter per minute change in flow for every millimeter we change the height of that stage. And we calibrate this at the fastest flow rate using a conventional, well-calibrated commercial system. So that's where we get 5% uncertainty at 1 microliter per minute. So how does the measurement work? We are delivering light across fluorescein here. And as fluorescein is exposed to that light, it's going to photobleach. And like I said, in this case, that's a phenomenon that we take advantage of. And the photobleaching rate is directly related to the intensity of light, how many photons are hitting the molecules moving through that region, and how long the molecules are in there. And that's related to the inverse flow rate. So very conveniently, we set up a dosage relationship here. So now we can set a fixed flow rate, scan through laser power, and map out the fluorescence intensity associated with a particular dosage. Then we can do the interchange at a fixed laser power, measure a fluorescence intensity, and back calculate what is the flow rate. So that's what we do. We step this controller through all of the various heights. We scan through all kinds of laser powers and basically demonstrate that we can reproduce this relationship over all combinations of laser powers and flow rates from 1 microliter per minute all the way down to 1 nanoliter per minute with 5% uncertainty. So what's really amazing is we've taken this calibration, which has 5% absolute accuracy at 1 microliter per minute, and scaled that relative uncertainty down by almost a factor of 1,000. So we've actually improved the accuracy of the measurement by 1,000-fold. So effectively, we can take any best measurement of flow and through this transformation, make it better using light. And I'm going to talk more about the optical profile improvements that we've made, but basically by creating this uniform light profile inside the microfluidic device, we get really sensitive measurements of flow around 0. So this then now becomes the world's best leak detector because we can detect changes in flow in just a few picoliters per minute. And this is, again, demonstrating the accuracy recalibration down to 1 microliter per minute, or 1 nanoliter per minute. And just to show you the demonstration here, we're taking 50 micron steps of this water column, and you see on the left measurements from the upstream fluorescence detection and the downstream fluorescence. That gives us the polarity of the flow. And on the right, you see the world's best commercial flow cytometer or flow meter showing no apparent change. So effectively, these steps are 30 picoliters per minute in real flow volume, according to the calibration of the conductance of the system. And this is in real time. So we're making really accurate, really quick measurements of flow within this system. So one of the questions you might ask is, what about the dynamics? Because we're looking at the interface where confusion and advection, or diffusion and advection are balanced. And so diffusion is going to play a role here, as well as how long does it take molecules to move through the flow meter at a particular flow rate. So we had this question of understanding the dynamics and looking at the time constant of the response. And my colleague, Paul Petrone, who is a brilliant mathematician, worked out the math, basically showing that this really brilliant relationship that you can calculate any time constant based on a time constant that you've measured at a particular dosage simply by scaling it by the change in flow rate. It's a really beautiful finding that we published last year So now you can understand how to utilize the laser power, which is one of your dosage controls, to work in a regime where you can set your time constant. So if you have a demand for a particular fast time constant, you set your laser power such that you can work in that dynamic regime. We also had some fun with this. We wanted to see what we could do with this. So we hooked our flow meter tubing up to a vortexer and turned it on. And you can see how fast it can measure the dynamics of flow here. Here you can see the optofluidic flow meter measuring all of those unique mechanical vibration frequencies within that vortexer. And here's the thermal flow meter showing you that it basically has a frequency cutout here. It drops out, it drops below the measurement of the actual flow rate, which is 100 nanoliter per minute. It picks up some of the dynamics, but it's got a frequency response that's considerably damped. And here again is looking at that in more detail, comparing a few different flow meters to the optofluidic flow meter, which is the blue trace here. So we're holding steady at about 100 nanoliters per minute, turning on that oscillation. And if we compare the frequency response of that, you can see that the optofluidic flow meter pulls out that frequency of oscillation quite precisely. Even if we speed this up to 3,000 RPMs, we get a frequency of 50 hertz, which 50 times 60 is 3,000. So it really is a sensitive mechanical sensor of dynamics in the flow. And we've now used this to look at the accuracy of syringe pumps. So we have syringe pumps with different fidelity. Here I'm showing you a low precision syringe pump, which is a really rough screw. If you've seen these, a lot of times these are standard undergraduate and graduate syringe pumps. With the optofluidic flow meter, you can see here, we can see every single click of the motor on those syringe pumps. And we can look at the inaccuracy based on the setting of the other two commercial flow rates as well. So here we're setting it 20 nanoliters per minute, and it's measuring a little high, so the syringe pump in this case may be a little bit inaccurate. And we can do that too for a more precise syringe pump. So this is something more like a Harvard apparatus syringe pump, which has a much finer tooth comb. Again, we can see all of those little clicks of the syringe pump, but the accuracies here are pretty good. So I can say that a medium resolution syringe pump does pretty well down to a few nanoliters per minute. And if we now compare low, medium, and high resolution syringe pumps here with the optofluidic flow meter, you can see that sometimes the high resolution pump, these scale too in about a factor of 10 in cost. So the low resolution syringe pump, maybe a few $100s. The medium resolution syringe pump is going to be a couple thousand, and the high resolution syringe pump is more than $10,000. So, here you're thinking about, am I getting what I'm paying for? Maybe, maybe not. The medium resolution syringe pump seems to be operating quite well in this regime. So, just show you more measurements here. I'm just going to move through these. But what's neat about the optofluidic flow meter is, of course, if you set a tolerance, for example, of 10% variation around your desired flow, you can see how much of the flow delivery that you're trying to accomplish is actually falling within that range. So I propose to you that almost nothing that you're trying to control at 2 nanoliters per minute is within any reasonable specification of that. So what are we learning with the optofluidic flow meter? We can use the photobleaching of molecules to encode dosing of the dopant in the liquid stream, and use that to scale the flow rates from a calibration down to the same relative accuracy in a much smaller regime. We could do dynamics also about 100-fold faster in time. And we can use this, my colleague Paul Patron has demonstrated in a couple of theoretical papers, that what's really amazing about this technology too is that the measurement performance doesn't depend on any particular channel geometry. It doesn't depend on any particular resolution in any features. It doesn't have to be rectangular sidewalls. it can be round, it can be triangular, it can be whatever it is, as long as it's consistent during the measurement process. It doesn't matter what the laser profile is. It can be Gaussian, it can be uniform, it can be a gradient in one direction. And it doesn't matter if the flow profile is laminar or plug flow. All of these different conditions work just as well for measuring flows using this technology. All right, so we're going to jump to part 2, which is flow cytometry. So a device that we've been developing to understand and improve the measurement of individual particles in flow. So a flow cytometer, as you know, is a well-utilized clinical and biotechnology instrument to characterize the heterogeneity and abundance of molecules on single cells and particles. The way the technology works is you have a single cell that has different antibodies that recognize different antigens on a cell surface. You flow them single file past a laser. You collect the fluorescence emitted by each of those unique labels, and that gives you the ability to estimate the abundance of that particular biomarker associated with each individual cell. However, because you're only measuring each object once and you don't understand the variability of the system, it's quite impractical to estimate the uncertainty of any particular measurement itself and to understand that measurement within the population that you're measuring. I'm going to get into this in more detail. So that effectively limits comparability of measurements on different systems. It makes classification subjective, and it also really prohibits measurements of rare events. So for flow cytometry, the signals that we are measuring are a convolution of the fluorescence shape of the object moving through a laser interrogation region and the shape of the optical profile that the laser is imparting on the cell. The way conventional instruments work is they measure this optical profile and convert it into 3 scalars: the integrated area of that profile, the height, maximum intensity of that profile, and the duration or width of that profile, and utilize that then to, for each object of measurement, put that on a scatter plot showing you maybe one biomarker versus another biomarker, or in the case of just scattered single color light that's not fluorescent transmission, you can estimate the size of a particle. So in forward scatter, that's typically used to estimate size. And so here now you can understand 2 different populations based on the characteristics of extracting those metrologies. But you might ask, well, how does the shape of one thing compare to the shape of another thing if you're not sure those things are passing the laser interrogation at the same speed, or if one of them is closer to the laser than another, one of them is smaller than the laser? So you really don't understand the operation of the system that gives you variability in measurement. And so really all you can say when you look at a scatter plot of cytometry is you understand the distribution of what you've measured, but you don't know how much of that distribution is due to the measurement itself and how much of that distribution is due to the inherent variability of the population you're being measured. And so in order to do single particle classifications, you really need to understand what is the uncertainty of that measurement in itself. So that's the problem we set out to address, and we've done this by creating a technique called serial cytometry. And in essence, we're measuring each particle multiple times by replicating this interrogation region along a single channel. In order to do that, we have to solve several problems. We obviously want our replicate regions to be somewhat identical in their performance, but we also have to maintain the position of cells within the channels from one region to the next. Ideally, we want our cells all going the same speed, or we're going to lose track of them. They're going to pass each other and we'll never be able to match. We also have to think about cell fluid dynamic interactions, such as doublets, and how things might move around within the flow between one region and another. And we also ideally want to look at sample variations. And can we use signal information, not just scalars from conventional cytometry? Can we look at the full trace to understand more about the measurements that we'd like to make? So conventional cytometers use a process called hydrodynamic focusing. You essentially take your sample fluid and you surround it by a sheath flow in order to focus that sample fluid into the center of the stream. So we thought that must work, right? Unfortunately, it doesn't. And this has been somewhat ignored by flow cytometry for more than 30 years. In fact, if you have particles in a 100-micron channel moving at a meter per second, the inertial forces are such that very quickly these particles move off of that streamline. Now, this works pretty well for traditional cytometers because your measurement region is quite close to the focusing region. But it's within just a few millimeters that this starts to fall apart. And you can see this in real time in a true flow cytometer if you increase the velocity of the flow. And by increasing the velocity, you're increasing this inertial effect. And so you can see this population fall apart and even sort of fall into these unique two populations. So what's happening? Like I said, we have a three-dimensional focusing system here. So the one we built microfluidically, we're able to bring fluid in from the top, fluid in from the bottom, and from the left and the right. So we can put the particle anywhere we want in the cross-section of the channel. If we put it in the center, you can see we've dyed the core red, but the beads are green. And so very quickly, the beads are no longer with the core. They've moved off to the sides of the channel. And you can see that over a distance of just 2.5 millimeters from the focusing region, those particles are quite distributed into these inertial nodes. So if you think about the conditions within which this is happening inside a microfluidic device, we often use the Reynolds number to describe the ratio of inertial forces to viscous forces. And if you calculate this for water in a 100-micron channel, typical, flow cytometers are operating at meters per second. Here, using only 0.1 meters per second, you're already at a Reynolds number well above 1, which indicates inertial forces are significant. And the way to think about that is you have this gradient profile of velocity, which gives the particle lift, moves it towards the wall. But then you've also got wall repulsion forces, which are keeping it a little bit off the side. So it's finding some stable position within a rectangular channel. And we can do some cool things because we've also got our cytometer sitting on top of a fast microscope. We can look at if we have a sample fluid that has contrast in it, we can actually see the particles spinning and mixing up the fluid. And we can also look at interparticle interactions. So there also is a distance over which particles that are separated will exert forces on one another and actually push each other apart. And this is something Dino De Carlo described a number of years ago, which can lead to crystallization. So if you can put in particles randomly, but after a certain distance under inertial forces, they'll actually be crystallized into almost perfect spacing, which turns out to be a benefit in serial cytometry. And so our solution to this whole problem is just to pre-focus the particles to this stable position and to utilize feedback in the measurement to find that position quite accurately. And by doing that, focusing to that position, you can see now we've locked the particle velocities into a really low variation. So these particles are really all moving the same velocity and staying pretty much the same distance apart over many centimeters of flow through this microfluidic device. And just to give you kind of a view of how this looks in cross-section from the view of the particle, you can see if the sample is in green, that's where the sample is that's where the sample with the particles in it is when it gets to the optical measurement region. These are all images of dyes in solution too. This is a really cool experiment Matt DeSalvo did to visualize these flows. And again, you can see this in an expansion region downstream of the chip. If you've center focused, you see your particles are spaced all throughout this region. If you get close, they're pretty close to locked in, but once you really lock them into that node, you can see how uniformly spaced they are. And even at a really high density, as they were in this case, the crystallization is quite beautiful. So they're all really lined up and staying right where they are. And to the point of the time of flight measurements that we're able to make as a particle moves from one region to the other, we can actually measure the separation distance between two particles from position 1 to position 2 within the device. And we can actually see that particles that are within about 150 microns, they're exerting a force on each other that's moving them further away from themselves as they get closer to that second region. So we can actually measure the change in distance between two particles between just a few millimeters of flow, which is really amazing precision. And just to say that the velocity characterization that we're doing here gives us the ability to track quite precisely. So, we can track to within 0.5% loss of particles from multiple regions through the cytometer. So now that we can match measurements, we can normalize their measurements and calculate a coefficient of variation to give us an estimate of the precision of that particle's measurement. And so now we can plot for each particle's intensity, how well did we know that particle's intensity? And then going back to a traditional scatter plot for cytometry, not only can we see the intensities of the distributions that we measure, but we can actually put a boundary around how well we know each particle's intensity distribution. And we can modify the control of the instrument to understand how well it's replicating the measurements. So if we are center focused, the distribution of fluorescence intensities is a mess. If we put them on the inertial node, we very carefully reconstruct the distribution of the population. We can actually speed up the flow and maintain the distribution of the population. So this is also the only cytometer in the world that gets better at faster flows. That's pretty cool. And then we can control the ratio of the sheath to core, the size of the sample, the amount of sample going through volumetrically through the system. So we can increase the volume of the core up until the point that that core crosses the center line, and then you see the measurement variations start to increase. So what we've learned so far is the faster flow is okay, doesn't impact the quality of the measurements up into quite a bit beyond where one would expect breakdown of inertial forces. Center focusing is the worst place to focus the particles in the channel. And we can increase the performance of the cytometer, wasting much less sheath fluid and still get the same quality of the measurements by doing this process. We can compare our measurements to conventional cytometers now and show that we are close to the same sensitivity for limit of detection as a commercial instrument. But we can show you something that no commercial instrument can, which is the uncertainty as a function of intensity compared to the population distribution uncertainty. So we've been able to improve the ability to resolve single particles from different populations. And again, here's just showing a scatter plot from all the measurements of a multi-peak bead set, where here we've got 9 different intensities of beads scaled about a factor of 3 from each, all having the same size. And you can see what their distribution of uncertainties is as a function of the number of fluorescent molecules bound to those beads. The second thing that we've worked on, well, the first thing, inertial focusing, actually solved the second problem, which is the velocity control for matching. The third challenge is then really working to improve the optical profiles that we generate inside these chips. We started out very roughly with just a straight waveguide with a small lens on the end here to try to focus a Gaussian profile across the channel. We improved that by collimating the light through a tunnel. We lose a lot of light doing this, though, so it's a very nice uniform laser profile, but it's dim. And finally, really great postdoc Jalal Sadeghi came up with the idea of using a diverging profile to create a uniform laser profile without loss of light. And effectively how that works is from every reflection of light on the wall of this channel, you're straightening out the light. So you have light coming out in a Gaussian profile of angles from the exit of the optical fiber into the waveguide. But as that light propagates down the waveguide, it's actually straightening out. And by the time it gets to the end, we have a nice uniform optical profile. And so we've been able to create these nice flat top laser profiles, which gives us also additional measurement advantages, because now if you have this steep profile, you can use the entrance and length of this profile to measure the velocity of the particle and the size of the particle within one measurement region. Matt DeSalvo has now done simulations of these to show that we can both simulate and experimentally achieve almost any optical profile we can imagine. And he's done some optimization to predict where these smooth spots in flat top profiles are based on the angle of divergence and the length of the waveguide in the channel. And so you can see that at different distances down the channel, we get these repeat interference patterns such that we get these nice uniform laser profiles. What's also nice about these laser profiles is because the light is collimated, we can use the transmission of light, or the drop in intensity of light as a bird is flying in front of the sun, if you will, to estimate the size of the bird, right? Its drop in light is proportional to the drop in intensity. But the collimation gives us an additional advantage in that we know that we know exactly when the bird starts interacting with the light, because that's the moment the light starts to drop. And that's an index of refraction independent measurement of size. So the way typical flow cytometers work is they use the scatter of light due to the index of refraction difference between the particle and the liquid within which it's flowing to measure the diversion of light on the edge of the particle. So it's very difficult to calibrate the dimensions of a particle in traditional flow cytometry. Here we can do it directly in a way that doesn't depend on the index of refraction. And we've done this now for different sized particles of polystyrene. We've also got glass and latex to validate this measurement. But you can see that we can project the diameters down to 0, again, which is something you can't do with a traditional scatter technique. Thinking now about the shape of the profiles, like I said, again, the measurement of the shape is the convolution of the laser profile and the distribution of molecules in the particles itself. So if we measure a bunch of particles, we might think that there's a way in which we can constrict these dimensions to understand how the particle interacts with the profile. And so if you think about a particle moving through the laser at one velocity, if it's moving faster, that's going to compress it in the time domain. If it's a smaller particle, that's going to compress it both in the time domain and in the intensity domain. Or if it has a different concentration, it's going to shrink in that way. So Paul Patron has come up with a method to do frequency analysis, spectral analysis of these signals, such that he can do these transformations, collapse all the signals onto a master curve, and estimate what were the changes in radius, concentration, and velocity that were responsible for those differences in distribution. And so we can effectively back out what was the distribution in particle sizes and velocities that were dependent on the conditions in the flow, and predict even a reduced uncertainty from what I can show you from the repeat measurements themselves. So we've done some work to validate this by changing velocities. So we just ramped our sample flow rate here from a high flow rate to a low flow rate, and we measured all these particles moving through in time, and we did the transformation analysis. Again, you can see how well the extracted velocity matches the time of flight velocity that we're already measuring. Again, here we're extracting the velocity from within one region using its shape. And then again, because we've normalized the velocity, the question is, well, we know they're the same size. Does the program predict that the sizes were maintained? And it does. We've also done this, I'm going to speed up here a little bit. We've also done this with particles of different size, and also looking at concentrations to show that those normalizations also make sense. And now we've applied this to mitotic cells. So by labeling the DNA concentration in the nucleus using a Hoechst stain, normally you use this to estimate the mitotic index of a cell. So cells that haven't divided their DNA are in G0, G1 with one copy of the DNA and one copy of the fluorophore, versus cells that have gone through synthesis now have twice as many labeled DNA molecules. We can look at the shapes of those and do the transformation to normalize all those shapes, and then back out what was the effective radius of the nucleus of those cells. And it turns out that that ratio is 1.25. And if you do 4/3 pi r cubed, where r is 1.25, you get 2. So it's a nice validation that a cell that is dividing approximately doubles the size of its nucleus right before it divides. Because we're repeating measurements in the channel, we can also ask questions about making measurements in time. Everything I've showed you so far is assuming nothing is changing, but there's nothing limiting us from making measurements of things that are changing between measurement regions. The first project that we've utilized to take advantage of this is deformation flow cytometry, with Brown using particles that are of low elastic modulus that are better matched to cells. As those particles deform in flow, they take on different time of flight trajectories. And it turns out we can measure that, and we can see pretty small changes in elastic modulus. So just a factor of 2 elastic modulus gives us almost complete separation of particles of the same size. We also thought we might be able to detect changes in the shape of the signal, and although it's there, it's not particularly useful from a metrology perspective, but it's something we were hopeful until we saw it. We're also putting droplets in these devices, so we are now controlling the concentrations of dyes and substrates to create reactions in droplets. This is a nice system because now by controlling the concentration of the fluorophore, we can actually use this to calibrate the measured fluorescence intensity of our cytometer. Normally you have to do that with plastic particles, but again, those particles are embedded in a matrix, and does that matrix give you the true fluorescence intensity of a dye bound to a cell? It's unknown. So here we have the ability to calibrate to fluorescence intensity, and we can scan through these different concentration distributions from a full gradient within a minute. We've utilized this in a chip that has time delays between measurement regions to get about 10 seconds or so measurement delay within a system. And we introduced a beta-galactosidase reaction where we control the concentration of FDG fluorophore reagent that's broken up by beta-galactosidase to create fluorescence in the flow. And you can see within these droplets, as they move from region 1 to region 2, 3, 4, 5, we're seeing that reaction producing fluorescein. And then we can characterize all of those droplets one at a time because they're all matched. In a droplet by droplet basis, look at the kinetics of that reaction through a scan of concentration that only took a minute. Really nice demonstration of droplet microfluidics. And we're now using this to look at polymer coacervates, so understanding the aggregation and accumulation of materials in polymers based on conditions that cause them to condense or expand. So we're looking at scanning out the coacervation space of 3 components of salt, polymer, and the flocculation reagent to map out the phase diagram of these novel coacervation reactions in real time. So what have we learned from serial cytometer? We've learned that repeating measurements gives us the ability to estimate the per particle uncertainty, which is independent from the population from which you're measuring. So typically we understand uncertainty from a very narrowly defined standard, bead. How does that relate to the distribution of a cell's stain that you might be utilizing? So there you're comparing something that has less than 5% variation to something that may have 300% variation in a completely different matrix, so plastic versus a cell in solution. So still sort of untested how well reference materials really enable comparison of fluorescence measurements. Because we can repeat that now, we have the uncertainty on each particle independent of what it is. We can do on-the-fly optimization of the instrument control, and we can look at dynamic measurements. And we're also utilizing this to do label-free estimation of particle elasticity. We've shown that better flow control leads to better measurements. This is independent of serial cytometry. Every cytometer can do this and should do this. We've shown that by improving optical profiles and extracting the information from the signals themselves, you can get a lot more information about the material that you're trying to characterize. And now pushing this forward, where are we looking to go? As I said, we're working to validate the refractive index independence of size metrology and seeing how far we can push that down to uncertainty bounds. We're looking at expanding into bioactive nanoparticles, so The work that Steve's group is doing with RPS measurements is very interesting to us. And then thinking about how to do single particle or even single particle from single cell metrology, I think is a unique way to think about heterogeneity of the way these extracellular vesicles are secreted from within a complex population of cells. We want to try to get to cell dynamics, so looking at calcium signaling and toxicity. We've already started looking at membrane permeabilization through the kinetic serial cytometer. And we want to do continuous monitoring in line in a bioreactor-like setup, so where we can pull off samples from a bioreactor and understand how the bioreactor might be changing. And part of the reason why I'm here is to try to do all this in plastics. So we started this with the help of Maggie and Matt, and especially Poubidou, to transition from PDMS into plastics. So I showed up here this week with this PDMS template, and we cast this now in MD700 and replicated it in COP, and you can see we're getting there. Something is happening. We've got it shot. So the devices that I cast were about 130 microns, which I validated before I got here. Poubidou agrees with me that they're about 130 microns, and our replicas also fall in that range, so they will work very well for 125 micron diameter optical fiber, which is the part we need to transition to. So with that, I'd like to thank my team. I've got a great team of postdocs, especially Matt, who's moved on from postdoc. He's now staff in the group. Meg Catterton is finishing up her National Research Council fellowship, so she's on the market. She's fantastic, especially with 3D printing. Brandon had just joined the group in November, and he's working to get us up to speed with nanoparticles eventually. And great interactions with students from Brown, including Graylin and Nick. And Eric Esch is a grad student from the University of Maryland who works full-time in my lab. Great collaborators, especially Paul Patrone, physicist and mathematician, has done awesome work on developing the theory and analysis for the signals and the flow metrology projects. And with the coacervates, we've got Vivek and Jeremy, his postdoc, working on that project. So thank you, Kansas. Rock chalk, and I'll take questions.